Hi everyone. Today we are going to talk about the chapter 4 and I'm going to go over the part 1 lecture presentation for AP1. So in this chapter we are going to talk about different types of tissues as well as where they are located and its functions. So tissue we can define as a group of cells which performs a similar structure with related to a common function. And histology, the word defines the study of tissues. And when you think about the types of tissues that the body is being made of, there are four basic types tissues. They are the epithelial tissues, the connective tissues, the muscle tissue, and the nervous tissue. And we are going to go over one by one. To view human tissues, because they are very microscopic structures, you need a microscope, and also because they are and there's no color on it, we have to first prepare the tissue in order to view it under the microscope. So there are several steps involved in this process where you first fixed the tissues. That is, fixing means tissue is preserved with some chemical solvent and it's been sectioned and cut into slices. By fixing, the tissue get little hardened enough where it enables us to cut. So in sectioning, you made lots of tissue slices, but they're very thin, and the to be very thin because it needs to be enabled the light or the electron beams to pass through that tissue in order to view its components. And then if you're using a light microscope, you need to stain them to enhance its contrast as well as to differentiate its several organelles, and therefore you use some color dyes onto it. But electron microscopes, they use some heavy metal coatings because they use the electron beams. So let's talk about the epithelial tissues first. The epithelial tissue, or we call it as epithelium, is a sheet of cells which covers the body surfaces or cavities. There are two main forms. There could be either covering and lining epithelia, or it could be the glandular epithelia. Glandular means it does have glands, so it does perform some function of secretion of certain substances. The main function of epithelia would be the protection, absorption, filtration, excretion, secretion, and sensory reception. So you need to know this in order for your exams. How we identify epithelia? By identifying its distinguishing characteristics. Actually, there are 5 major distinguishing characteristics, and these are common for all epithelia. There could be little variations, but almost all epithelia does have these characteristics functions or characteristic features. The first one is the polarity, and the second, specialized contacts, and it's supported by connective tissue. The avascular, there is no blood vessels, but it's innervated by nerve tissues, and the regeneration ability. So polarity means that cells have a polarity, which means like it does have a top and a bottom. And the top part, we call it as the apical surface, which is the upper free side, and it's exposed to the surface or a cavity. For instance, like the skin cell, the epithelia, which is the stratified squamous epithelia, it's been exposed to a surface, like the outer surface. But we'll think about the lining epithelia of the internal organs. They're exposed to a cavity of that internal organs. The apical surfaces, they are very smooth, or specialized finger-like projections based on where it's located, because not all apical surfaces have finger-like projections. If they do perform functions such as like movement of substances or in order to absorption process, which increases the surface area, then it does have some finger-like projections. The basal surface is the bottom surface, which is attached to uh, the lower part, and it faces inwards towards the body, and it attaches to the basal lamina, which is an adhesive sheet that holds the basal surface of the epithelia cells to the underlying cells. So it's like a little base which is under the epithelial cells. The specialized contacts is another characteristic feature of epithelia, because epithelia tissue needs to fit closely together, and the specialized contact points bind adjacent epithelial cells together. So if you do remember, we have learned in chapter 3 different junctions. So those are type of contacts that epithelial cells do have. The other important characteristics would be the connective tissue support. So all epithelial sheets are supported by the connective tissue, and it does have a reticular lamina, which is the reticular lamina located deep to the basal lamina, and the reticular lamina consists of network of collagen fibers. So sometimes when you look at the histological pictures, you would see like reticular layer. So at that point, you have to remember this reticular layer means it's a type of connective tissue, and it does have collagen fibers. The basement membrane is also the connective tissues, provide connective tissue support. So the basement membrane is made up of basal and reticular lamina. It reinforces the epithelial sheets. It resists stretching and tearing of the cells, and it also defines a clear boundary for the cells. So don't forget that it's a combination of the basal lamina as well as the reticular lamina, the basement membrane. So don't think it's just like one structure. It's a combination of the underlying connective tissue as well as the basal lamina. Let's look at the clinical relevance of what we have learned so far. So cancerous epithelial cells, we know that it's such a problem with related to health, and they do have like ability to creep through different cell layers. So why is so is that they do not contained by the confined or contained by the basement membrane boundary. So they can penetrate the boundary and invade the underlying tissue, and that will result in the metastasis, which is the spreading of the cancer cell from its primary site. The other important clinical feature would be the avascular. So there is no blood vessels found in the epithelial tissue, but some sort of nourishing needs to be happened. So that is happening through the diffusion. Remember, it's the diffusion, the definition, passing molecules from higher concentration to the lower concentration. So it's happened from the underlying connective tissue. The nutrients comes through the diffusion to the epithelial tissues. But at the same time, it's been innervated by the nerve fibers, so it has a good supply of nerve fibers. The next important function would be the regeneration capacity, because epithelial cells have a high regeneration capacity, and it's been stimulated by the loss of apical base polarity and broken lateral contact. So for instance, if you have a wound or if you have a scratch, the top layers would be take off due to the injury, and then the lower cells, the epithelial cells, would able to divide and repair the wound area, so that it does have the regeneration ability. So some cells are exposed to friction and some hostile substances, and that would lead to the damage, and that is kind of the stimulus removing of the cells, as well as stimulus for the start of the regeneration. So based on several features, you need to understand how we name the epithelial tissue. So classification of epithelia is done due to several features, and let's look at them now. So if you look at an epithelial tissue, how it has been named, you would see there are two components. You have a first name and you have a second name. So the first name, which is like either simple or stratified, is indicated by the number of cell layers. So if you have one single layer of cells, you called it as simple epithelia. If you have two or more layers of cells of thickness, then you call it as stratified epithelia. The second name is, of course, due to its shape of the cells. So there are only three shapes. It's easy to remember. It could be squamous, which is the flattened and scale-like cells, and it could be cuboidal, which is either box-like or cube-shaped cells, or it could be columnar, which is tall or column-type cells. So if a stratified epithelial shape can vary in each layer, then you label the stratified layer based on the apical cell surface. So repeating once again, because you might have seen some stratified cell epithelia, especially the skin. Not all cells of that stratified layer has the similar type of cell shape. Some of the cells, which is at the basal layer, would be cuboid in shape. But as it moves on, the cells get flattened and getting much more scaly type. So therefore, when we have to name them, that is what has been sent at the bottom, when we have to name them, what we take is the shape of the apical cell layer. Apical is the top part of the cell layer, and then you named it as it is. For instance, the squamous type cells are in the skin epithelia, even though it has several layers down below. So we call them as stratified squamous epithelia. Like that. So you look for the top cell layer when you have more than two cell layers to name the cell. So here we see the stratified and the simple description. So if you see the basal lamina, the basal surface of the cell, and this is much more flattened cell. And of course here you say like it's one single layer. So you see it's a simple epithelia, and you do see the nucleus over here. And of course in this figure you would see there are several layers. This is a first cell layer, basal cell layer, the top cell layer, the third one, fourth one, and the fifth one, and this is the apical, the top cell layer, and you would name this based on the shape of this apical cell layer. And here it shows like how would be a squamous cell look like from the top view and from the lateral view. And from the lateral view, it would be cuboid. It would be much more flattened, scale-like, and this would be much more column in shape. So let's look at each type of cell epithelia and their functions, where they're located, and this would be a very good area for your multiple choice questions. So simple epithelia, of course, is a one single layer, and it's involved in absorption function, secretion function, or filtration processes. And simple squamous epithelia, they are cells flattened laterally, and the cytoplasm is sparse on this. So here you would see the flattened cells with disc-shaped central nuclei and some sparse cytoplasm, and this is the simplest of the epithelia. So the functions would be to allows materials to pass by, diffusion and filtration happens, and also it can secrete lubricating substances in the serosa. And you would encounter these simple squamous epithelia in kidney glomeruli, air sacs of the lung, lining of the heart, blood vessels, lymphatic vessels, lining of the ventral body cavity, which is the serosa. So you need to remember actually this whole figure to in order to perform your multiple choice questions very well. Function, location, and its special features. The next one would be the simple cuboid epithelium, simple one single layer cuboid. The cells of that cell layer would be in cuboid in shape for lateral view. It involves with secretion and absorption process. It forms walls of the small ducts of the glands and many kidney tubules. So any gland, the duct would be from the simple cuboidal epithelium. And the locations, kidney tubules, ducts, and secretory portions of it, and some small glands, ovary surfaces, you would find this simple cuboidal. And when you look at this, actually this is a cross section of a duct. So it's like when you take a pipe and cut it and looking from the top of it. So although the cell demarcations, the walls, the margins would be not clear, if you look at some of these cells, for instance, like where my arrow is pointing, you do see a cuboid in shape, and you do see a well-round, nicely located nucleus in that cell. So even though, like, based on where it's been cut, you might see a nucleus of that cell or you might not, like for instance like in here. But if you grab the nucleus when you cut it, then you would see the nucleus, nice round shaped nucleus. So based on the number of nuclei you see, you can tell how many cells you have in that duct as well, in that cross section. But you don't necessarily need to know how many cells in there, but you need to know how to identify. So look for the cuboid shape of the cell of that figure, and to figure out whether it's simple cuboidal or simple columnar. So the simple columnar, on the other hand, it's a single layer of tall, closely packed cells, and some cells, they do have microvilli and some have cilia on top of their apical surfaces. And also some layers, they do contain some specialized cells called mucus secreting goblet cells, and especially you do see them in intestine epithelia. So they do involved in the absorption and secretion of mucus, enzymes, and other substances. The cilia cells, they move the mucus on top of their apical surfaces. As I mentioned before, the simple columnar mainly found in the digestive tract epithelia, the gallbladder, ducts of some of the glands, respiratory tract, the bronchi, as well as in the reproductive system, the ovarian tubes you do have. So think about the functional relationship, because bronchi, you need to sweep off the debris which you get from the air out of your airway. So that does have the cilia, and the microvilli, they do have a function for that. The uterine tubes, they have to sweep the ovum, the egg, towards the uterine cavity. So relate your function into the structure, and that's you can, and it would help to remember for the digestive tract. Microvilli helps to absorb nutrients as the food passed through it. The gallbladder, same as for secretion purposes and for mucus purposes. Yeah, here you would see the simple columnar epithelia. So here you see a cavity, this white space, you see this little, you don't see it much clearly nicely, but here this little layer would be the microvilli. It's like each would be representing one single microvilli. So there are thousands of microvilli on top of this. So this is the apical surface of the cell. And look at this boundary. You do see a column-shaped cells over here, and you see an elongated nuclei. Okay. And this is the basal lamina, which is not obvious very clearly, but when this is the apical surface, you do find it as the basal lamina over here. The next one would be the pseudostratified columnar epithelium. The cells, they can vary in height, and it appears to be multi-layered and stratified, but tissue is in fact a single-layered simple epithelium. It's involved in the secretion, particularly of the mucus, and it also in movement of mucus via ciliary sweeping action. And they're mostly located in upper respiratory tract and ducts of large glands, and also tubules in testes. So pseudo means is false. So obviously it looks like a stratified epithelium, columnar epithelium, but when you look closely, all cells would lie on one basement membrane. So therefore, you do see it like it looks like as stratified, but it's not obvious. It's multi-layered. The appearance would be multi-layered, but in fact, the tissue is layered in the same basement lamina. So this is what how you would see like a pseudo-stratified epithelium. If you look at it, even though like how you say is stratified means like you would, if you see a stratified epithelium, you do see the nucleus in different locations. So you look like, it looks like a stratified epithelium, but if you look closely, all cells, like even though this nucleus lie in these cells at the top, this cell lies at this basement membrane. So all cells lie at one basement membrane. And you do see like single specialized goblet cells here, which involved in mucus secretion. And here, of course, you do see nice microvilli, lots of microvilli in the apical surfaces of these cells. So now let's look at the stratified epithelia. Stratified epithelia defines which is like two or more cell layers of the cell. So more durable than simple epithelia because it does have a lot of cell layers, and it does provide a protection as its major role. And stratified squamous epithelia is most widespread of stratified epithelia. Of course, if you think about, the skin is considered as an organ of our body, and skin is the largest organ of the body. So obviously, the stratified squamous epithelia would be the most widespread epithelia in our body. And the stratified squamous epithelia, the free surface is squamous, so that's why it names as the squamous epithelia, and with deeper cuboidal or columnar cell layers. That it located in areas of high wear and tear, example is the skin. And some do have keratinized cells found in the skin, and there are non-keratinized cells are found in most linings, because inside stratified squamous epithelia, you don't find keratinization, but outside in the skin, you do find keratinized cells. So here it shows the stratified squamous epithelia of the skin. You would see like much more cuboid shaped at the bottom, and as it moves on, the cells get much more flattened, and you do see some keratin cells, and the top cells would be much more keratinized in this cells. And the non-keratinized types, they found in most linings of the esophagus, mouth, vagina, and the keratinized, it would be found in the epidermis of the skin and also the dry membranes. The stratified cuboidal epithelia, they're found in some sweat and the mammary glands, and typically they do contain only 2 cell layers in thickness. And the columnar type, they're very limited distribution in the body. You would find small amounts in the pharynx, in male urethra, and lining some glandular ducts. And usually you would look for those stratified columna in the transition areas between 2 other types of epithelia. Um, for instance, like when you transition from the esophagus into the stomach, you have a transition zone. So you might need to look for those areas for stratified columnar epithelium. And one apical layer is column. Its only apical layer is columnar. So in that stratified columnar, you would see only the apical layer is the columnar type. The rest would be cuboidal in shape. There's another type of stratified epithelia called transitional epithelium. It forms lining of hollow urinary organs, which is found in bladder, ureters, and urethra. The basal layers either could be cuboidal or columnar in shape. And the characteristic feature of this epithelia is the ability of these cells of this epithelia to change shape when it's stretched. Because if you think about the urinary bladder, that it has to stretch when the urine is filled up, but has to go back into its normal position when the urine is pushed out. So it increases the flow of when the cells stretched, when there is increased flow of urine, and in the case of bladder, it does provide a storage space for that organ. So here it would shows the transitional epithelia. It resembles both the stratified squamous and stratified cuboidal. Basal cells would be cuboidal or columnar cells, and columnar shaped. Surface cells dome shaped or squamous like, depending on the degree of the organ it's been stretched. And the function would be obviously it stretches easily and it permits stored of urine to distend the urinary organ. And mainly you would find in the ureters, bladder, and part of the urethra itself. The other important features we need to talk about certain other features in the epithelia, which is like sometimes the gland. So gland is defined as what gland we can describe as there are one or more cells which makes and secretes an aqueous fluid called a secretion. So gland, it could be single cell gland, or it could be like multiple cells involved in this glands making. So glands we can classify by the site of product release. Either it could be an endocrine gland or an exocrine gland. The endocrine glands, they're internally secreting substances. Examples would be the hormones. The exocrine glands, they externally secreting secretions, which is example of sweat glands. Sebaceous glands, they excrete things outside. And also we can classify by the relative number of cells forming the gland. Either it could be unicellular, which is formed by one cell. Examples would be the mucus secreting goblet cells. The multicellular would be the salivary glands, which secretes the saliva. The endocrine glands, they are ductless glands because a gland, it should have a glandular component as well as a duct. A duct means a pipe-like structure which takes the secretions towards the outer surface. But endocrine glands, they do not have this pipe component. They do not have the duct. So we called it as ductless glands. And the secretions, they are not released into a duct, they're released into the surrounding interstitial fluid, and that secretions is being picked up by the circulatory system and carried into other sides of the body, and secretes hormones by exocytosis process. They secrete messenger chemicals, as well as they travel through the lymph or the blood to their specific target organs. And based on the receptors that the target organ has, the target organs respond into its specialized characteristic way. The exocrine gland, the secretions are released onto the body surface or into the body cavities. So more numerous than the endocrine glands, they secrete products into the ducts, and examples would be the mucus, sweat, oil, and salivary glands. So it could be unicellular or multicellular, as I mentioned before. Here you would see the types of the glands, the simple duct structure. You would see the duct is this yellowish color portion. The glandular portion is being shown in here. So simple tubule, it's almost like one tube. So the glandular part has not been divided. So examples would be the intestinal gland. Here you would see simple because the duct has not been divided. And it's been branched. The glandular part has been branched. So we call it simple branched tubular glands, which is you find it at the stomach and the gastric, which are the gastric glands. And here you would see the simple alveolar gland. It's the shape of the glandular portion is like much more rounded and alveolar in shape of the lungs. So here it's a branched alveolar. Obviously, it's you would see the shape of the sebaceous glands would be this type, because it's simple. The the duct portion has not been divided, but the glandular is being branched. But the shape of the glandular portion is much more alveolar. Here it shows like the compound glands. Obviously, you would see the duct portion has been branched, so there are lots of branched glandular portions have been combined together, and therefore it could be either compound alveolar glands or the compound tubular glands, or it could be a mixture, compound tubular alveolar. So most of, like this, for instance, here this is the tubular portion of the gland, this is the alveolar portion, this is a compound tubular alveolar gland, like that. So with related to the multicellular exocrine glands, there could be based on the mode of secretion, we can divide them either into merocrine glands, the holocrine glands, or the apocrine glands. So merocrine glands, they secrete most products by exocytosis, and the secretions would be formed from the sweat glands as well as the pancreas. The holocrine glands, they accumulate products within the gland itself. The entire secretory cell can rupture and release its secretions and its fragments. Examples would be the sebaceous oil glands. On the other hand, the apocrine glands, they accumulate products within, but only apex cells, apex glandular portion get ruptures, and we don't know yet like whether this kind of glands exist in the human body. Some would think like mammary glands would be type of apocrine glands. So that is the example. The next important topic would be the connective tissues after the epithelial tissues. This is another major type of connective tissues, major type of tissue type in the body. So connective tissue is the most abundant and widely distributed of the primary tissues. There are four main classes: the connective tissue proper, cartilage, bone, and blood. So if you look at the characteristics like the epithelial tissue, you have to look for these characteristics to identify it as a connective tissue. So there are three characteristics make connective tissues different from other primary tissues. That is, all have common embryonic origin because they all arise from the mesenchymal tissue. Um, and they have varying degrees of vascularity because cartilage is avascular, but on the other hand, bone is highly vascular. So they have a different degree of vascularity. And then the cells, they are suspended or embedded in the extracellular matrix, which is the protein sugar mesh you find in your tissues, in the connective tissues. So the matrix, they support cells so they can bear the weight, withstand the tension, and also endure the abuse. So therefore, it does have a function. So these are the three characteristics: the common embryonic origin, different degree of vascularity, as well as the cells of the connective tissue suspended in an extracellular matrix. So all connective tissue also have three main elements, that is the ground substance, the fibers it does have, and the cells. The first two elements, which is the ground substance and the fibers, they make up the extracellular matrix that you need to remember. The ground substances. Let's look at it. It's an unstructured gel-like material which fills the space between the cells. So the components would be of the ground substance is that the interstitial fluid, cell adhesion proteins, which glue for attachment, and proteoglycans, which are the sugar proteins. Protein and glycans are sugar component. That's why it's called proteoglycans. They're made up of protein core or large polysaccharides. The proteoglycans is from the protein as well as from the large polysaccharides. The chondroitin sulfate, the hyaluronic acid you find is some of the proteoglycans you encounter in the connective tissue. And water is also a component of the ground substances, and it's trapped in varying amounts, and based on how much water it's been trapped, the viscosity of the ground substance would vary from tissue type. Then the connective tissue fibers. So you have to remember there are three types of fibers provide the support for the connective tissue. They're the collagen, elastic, and the reticular. So collagen fibers, they are the strongest and most abundant type. So they are tough. They provide high tensile strength for the connective tissues. The elastic tissues, it network of long and it's thin elastin fibers, and it allows to stretch and recoil. So elasticity, stretch and recoil. The reticular ones, they are very short, fine, and highly branched collagen fibers. They are different chemistry and form from collagen fibers, and they do provide branching forms of network in the tissue. The cells, on the other hand, is important as well. It could be blast cells or the site cells. So when you see the blast word, you have to remember that it's an immature form of the cells, and they actively secreting the ground substances for the extracellular matrix fibers. And you would find several types: the fibroblast, chondroblast, osteoblast, and even though the name is a little different, the hematopoietic stem cells, they all come under blast. So fibroblast, they're found in connective tissue proper. Chondroblast, chondro goes with cartilage, so found in cartilage. Osteo word goes with bone, so osteoblast found in the bone. The hematopoietic stem cells in the bone marrow. Site cells. When you find the word site, you have to remember that it's a mature type. They're less active form of the blast cells, and they become now part of and helps the maintain of the matrix of the connective tissue. The other cell types in the connective tissue you would encounter would be the fat cells. That's a different types of cells which store the nutrients, white blood cells, the neutrophils, eosinophils, lymphocytes, and they do provide the tissue response to the injury. And also you can find the mast cells, which initiates the local inflammatory response against the foreign and the microorganisms, and well as macrophages, which are the phagocytic cells that eat dead cells, microorganisms, and also it provides function in the immune system. So if you look at this figure, you would see different types of cells, different types of fibers, the collagen fibers, the thick tensile fibers, the thin ones would be the elastic fibers, and then the branched one would be the reticular fibers. You do see fat cells in yellowish color, mast cells with lots of vacuoles, and the neutrophils with divided nuclei over here. You would recognize them. The lymphocytes, one single nuclei with the round shaped cells. The macrophages, all would encounter, and the number of cells or the type of cells vary depending on the type of the cells. So let's look at let's stop at this point and then we'll continue with the rest